The biosynthesis of nucleotides. Nucleotides are like beads. One by itself is pretty useless, but when we put together with many more, it can make many important things. When these beads come together, they form a chain where it can grow. This is nucleotide synthesis, where simplicity turns into complexity. In this video, we're going to address what nucleotides are, as well as how they come about and making up DNA, RNA, enzymes and energy sources, and eventually us. A nucleotide has three components: a pentose sugar, which can be either ribose or deoxyribose, a phosphate group which binds to the fifth carbon, and a nitrogenous base which binds to the first carbon through N-glycosidic bonding. So, what makes up a nitrogenous base? There are two forms of nitrogenous bases: purines, which are two carbon nitrogen ring bases which consist of guanine and adenine as nucleobases, and pyrimidines, or one carbon nitrogen ring bases consisting of cytosine, thymine and uracil as nucleobases. Some of the nucleotides' central roles include being the building blocks of nucleic acids, storage of chemical energy, carriers of activated metabolites for biosynthesis of coenzymes and metabolic regulators. Purines have two kinds, endogenous and exogenous. Exogenous purines are absorbed by the body through the foods that you eat, such as organ meat, alcohol, seafood and sweetened beverages, whereas endogenous purines are made directly by the body itself. Now we're going to discuss the pathway of nucleotides. The synthesis of nucleotides is controlled by the liver, either via de novo or salvage synthesis. First, let's evaluate the de novo pathway. The de novo pathway is split into two: purine and pyrimidine. Nearly all organisms synthesize purines and pyrimidines via the de novo pathway. Using PRPP, the de novo pathway enzymes build purines and pyrimidine nucleotides from scratch, using simple molecules such as CO2, amino acids, and tetrahydrofold. This route of synthesis has a much higher energy requirement compared to salvage pathways. 5 out of the 12 steps of the de novo purine synthesis require hydrolysis of ATP or GTP. Salvage pathways of purine, 3 purine bases are released. The guanine and hypoxanthine bases are activated from the HGPRT enzyme. Nucleotides GMP, IMP are dephosphorylated by the enzymes GMP and IMP, creating nucleosides, creating the full cycle of the salvage pathway, with all parts being recycled. The same process occurs with the adenine-based salvage pathways. Adenine is activated from PRPP before converting into AMP. The enzyme APRT is used to recycle adenine back into AMP. Salvage pathways of pyrimidines. Cytosine and uracil are phosphorylated by the enzymes CMP and UMP, creating the nucleotide. Thymidine nucleotides uses the enzyme thymine kinase. This catalyzes the transfer of the ATP to thymine, creating the formation of TMP. Every step in the conversion of UMP to CTP is essential for providing the cell with the specific precursor molecules necessary for the DNA synthesis. Thymidine is converted into DTTP through TMP through phosphorylation reactions, the important feedback mechanism that regulates cellular ATP levels.